Hello, welcome to Living Science. Dr. Amit Kumar Dinda, professor at AIIMS, is here with us today. Dr. Dinda is an accomplished researcher, has published more than 300 papers. He has worked in multiple very different disciplines of science such as nephrology, pathology, cancer, nanomedicine, tissue engineering, and is also a practicing pathologist at AIIMS. He is regularly diagnosing cancer. So today we'll try to find out how is that he's able to wear so many different hats. Dr. Dinda, welcome. We are very happy to have you here. We'd like if you could give us an idea of what research done in your lab.
Yeah, I'm fortunate enough to really get a chance to work in the different areas of research in biomedical sciences, and particularly in pathology and pathobiology. I did my MD thesis in platelet functions in diabetes. So that's a more of a hematological area of work. After that, my PhD I worked in cancer biology. It's more of a brain tumor, glioblastoma. And after that, I did quite a bit of work in experimental pathology, developing disease model in different areas. And I took over the charge of immunopathology. And so as a part I am still doing a lot of renal research and renal oriented research. And on top of it, I think in around 2004 or 2003, I started working in association with IITs, particularly understanding material and cell interaction. So we have quite a few work in the area of the biomaterials and cellular and tissue interaction. 2009, rather late part of the 8, I started working in the area of nanotechnology applications in medicine. And in the first project which I funded project I got on the macrophage targeting. The issue was very simple. I thought that there are a lot of infections which are intracellular infections, particularly in the macrophages, like tuberculosis, chalager, and so many things, so many others. So if we can produce a nanoparticle or nanocarrier in which you can load the drug and it goes specifically to the macrophages, then it will carry these high concentration of the drug into the cells, into the infected macrophages, and thereby killing them more effectively. So from there my journey started. And after that, we are now developing similarly in the carrier for amphotericin B. There, our target is to develop it for chalager and as well as systemic fungal infection. And we have developed a platform where you can load 4 different drugs or different molecules in single nanoparticle. This we are developing for tuberculosis, where 4 drugs are essential to treat, which has to be in a particular ratio. So we cannot mix the different batches of nanoparticle carrying different drugs, because it will never reach in a equivalent concentration at the site of action. So we have another project where we are developing these oral gene delivery system, and also the siRNA delivery system on which we want to target the macrophages, particularly in the atherosclerosis. Thereby we are trying to revert the fat accumulation so that they can cause the regression of the atherosclerosis. And finally, the one area which we are really got a huge success is the use of nanoparticle for developing single shot vaccine. I mean, we have picked up a polymeric system where once goes inside these antigen presenting cells, it can have a slow degradation over 4 to 6 months, and thereby it can continuously present this antigen. And this antigen also we can modulate to do a cross presentation, whereas it will be mostly presenting through MSC1. So this kind of system will be very useful if finally when it comes. We have already published and done work with tetanus, with tuberculosis. Now we are developing it for hepatitis B, and I'm quite sure that it can be later developed from other intracellular infections also, including viral infections. And apart from that, we have picked up few projects on application in the cancer nanotechnology, where we are applying photothermal therapy, and then sentinel lymph node detections, where the cancer spread can be detected in the lymph nodes. And also I think the one project which we are recently trying to develop for treating of rheumatoid arthritis, which is a very, very important and difficult area to do. There also I am tweaking my experience in macrophage targeting, because in inflammatory milieu, if you can alter this macrophage behavior by targeting with some modifier or some molecules, then maybe inflammation you can reduce. So these are to name few of this project. On the diagnostic front also we are developing nanotechnology based urinary biomarker detection. So, there are, I believe that the new avenues are coming up as more and more we are understanding the different kind of nanoparticle fabrications and their applications. So in future we can maybe work more areas also.
So how is that you targeting the macrophages? So you putting something on the surface of nanoparticles, or?
Yeah, basically we are trying to target some of the receptors of these macrophages. We are targeting mannose receptor because our initial studies showed that mannose receptor targeting doesn't cause much alteration in the macrophage function. And at the same time, in infections, particularly the mannose receptor upregulations are also described. And plus, I think mannose receptor targeting gives us another issue of, if we are working with the vaccine, then antigen processing is much better, particularly for MSC1. So we are doing with the mannose receptor targeting, but then we can do other targeting also. Other FC receptor or other receptor one can do. It all depends upon what exactly we want to expect from these macrophages.
So why is that MHC1 class 1 response is better? Because it's slow release or constant? What's the reason?
Yeah, we are what we have shown by our initial results is that when we the polymer loaded antigens is engulfed by these macrophage and dendritic cells, they usually come out of these the endosome, and so they degrade in the cytosol. So basically, when the cytosolic release of antigen is there, they are mostly they can go to the MSC1, and plus we have a delayed maturation of these endosome also. That means our particles stay in longer period in the early endosome, and already papers are there that the antigens released from the early endosomal maturation can be diverted more to MSC1. We are still exploring a lot of avenues to further understand and modify our system by antigen presentation.
Dr. Dinda, obviously we understand the success, reasons for success of targeted drug delivery system using nanoparticles, but what are the reasons that non-targeted drug delivery system or nanoparticles are equally successful?
Yeah, I think it depends upon in what particular situation we want to use these nanoparticles. When we want to really target a particular cell populations, obviously we want to do the targeting. But then situations like, say for example, in a particular cancer regression, where sometimes we want these nanoparticles to get accumulated inside this tumor area, where it can just go through what we say this EPR effect, that is the defective vasculature. From the defective vasculature, these particles can come out in the tumor area and accumulate in outside the extravascular space in the vicinity of these tumor cells. Now they will be in the tumor area itself, but they may or may not enter some of them into the tumor cells themselves. So if we do that, then we can accumulate a lot of these the nanoparticle in the tumor area itself without specific targeting the tumor cells. Now this has got a plus point also, because some of these the payload of whatever we are using, drug or biomolecule, they will be released if they are endocytosed by the tumor cells inside the tumor cells themselves, and they will be also released outside these tumor cells in the milieu, in the stroma, as they degrade. So you have got both kind of effect is there depending upon what you want. And certain situations, say for example, some of these products which are available now in cancer nanotechnology based the cancer therapy, and they are not targeted, particularly say for example Abraxane. So it is doing pretty well. So I feel that when we want to do targeting, it has to have a very specific purpose, and all our cases of targeting we always compare with a non-targeted one. So if we find that it is not that way giving us extra example, then obviously we don't want to do unnecessarily targeting also.
Dr. Dinda, how successful have been your efforts in treating hepatitis B using nanoparticle based drug delivery system?
The hepatitis B vaccination strategy project which I have taken over, that is because of one of the issue which is actually known in Indian condition. Now, Government of India has developed mandatory hepatitis B vaccinations for all the its populations, whole populations. Now, in the villages, particularly primary health center, for first vaccination, so it is given in 3 doses, 0, then in 1 month, and then in 6 months. Now, day 0, the first dose can be given to almost 80% of these populations, if not 100%. But then after 1 month, actually it's only 10 to 20% which comes, and then after 6 months, hardly 5% or 10% comes. So there's a huge challenge to really do mass vaccinations with all these booster dose and repeated dosing. So we thought, can we develop something where the single injections can develop a effective vaccination or immunity for long. And so with that motto, we picked up this hepatitis B, and there we are doing these antigen cross presentation, particularly for MHC1 upregulation, where it's a viral infection, so cytotoxic T cells will be much more effective than these antibodies. And also we are using a very slowly degrading polymeric vehicle, so that single injections, the antigen presentation can be prolonged for several months. So our initial result which we have published, that is on tetanus, so that is totally different from hepatitis B system. And also we have published one in ESAT6, that is to some extent similar to, not similar, but then we can say that that's a tuberculosis also has intracellular infection. So basically we are now have completed our the cellular interaction study and immunostimulation study from the peripheral human blood derived monocyte and the human CD4 and CD8 cells. Now our the guinea pig model we are going to start soon. So this will be further validated, but our earlier publication shows so good result. We are very hopeful that we will be getting a quite good response in hepatitis B also.
Dr. Dinda, you have explored targeted and non-targeted nanoparticle based drug delivery system, and also you have explored the potential of these nanoparticles in vaccine development. We are wondering, is there any dream project which you have not pursued yet? And it will be great if you could share it with us.
Yeah, the immunotherapy for cancers is the one which is really will be fantastic if we can develop properly. See, one of the major issue in the tumor immunity, as you know, that these tumor cells or cancer, they totally abolish or they hijack the normal immune system against these cancers. So what we are, that's why planning, I mean that is my dream, I have to see, though there are few of these trials are available, what we want, that we want to take out the peripheral blood of these cancer patients, particularly with the advanced stage of cancers, and where there is no conventional therapy as yet, effective therapy. And then we take out these peripheral monocytes, and then these monocytes we want to convert into dendritic cells. And in these dendritic cells, we want to challenge them with the tumor antigen loaded nanoparticle. Now, tumor antigen loaded, what I am planning is that this is the whole pure tumor cell lysate. So we get the tumor from the same patient, and then we purify the culture of the tumor cells, so there is no contamination of other cells, and then we make a lysate. Maybe we can try out something interesting, like we can make the tumor cell die by apoptosis, because it is known that if you give the apoptotic tumor cell lysate, there are some preliminary study that it can enhance these dendritic cell based antigen presentation. But then these are some experiment where it is in culture mode, these antigens are given to the cultured dendritic cells, but here we will be giving through our vehicle, which is known to have a very high potential of antigen cross presentation for MHC1. And then also, not that, I am thinking little ahead of that. I thought that if we can do so much, then is it possible that we can take some blood from these patients, isolate their T cells, and then prime in ex vivo conditions with these antigen loaded dendritic cells with the patient's own T cells, and make them, it is possible to expand the clone to some extent in vitro, and then infuse them together to this patient. So what will happen, these are all autologous systems, so I don't find any ethical issues, any regulatory issue. At the same time, the tumor which is suppressing the host immunity, it can be bypassed. So obviously it's a dream, because there is a lot many things to be done, and but I think maybe it's feasible.
Dr. Dinda, you talked about use of nanoparticles in cancer imaging. Can you expand it a little bit for us, please?
Yeah, the nanoparticle can be really multifunctional, particularly for imaging. When we talk about imaging, you can use the fluorescent imaging, or you can use the magnetic imaging. Now, the one which is really and also you can use some of these particles which have got a very high radio enhancing capability, say for example gold nanoparticle. But for MRI imaging, iron nanoparticles are being used. I mean, they are being developed now, and so all these imaging, it can really enhance and target even the cancer cells. So, for example, you can develop a iron oxide nanoparticle and tag a particular ligand or antibody against the cancer cells, and if you inject it intravenously, then it will go and bind to these cancer cells. So, in metastasis, early metastasis detection, it may have a role. So, even if in a routine MRI you can't pick up very small area of recurrence, but with this kind of antibody tagging you can actually detect them very easily. And similarly, this in vivo fluorescence imaging, which is also basically fabrication of these different nanoparticles with near infrared imaging, where your depth of these fluorescence, the light which can pass through your tissue is around 12 centimeters. So, this is quite deep. The major issue of all these nanotechnology based imaging system is to really prove that they are non-toxic and they are safe. Most of them has got metal nanoparticle like gold and iron oxide. Iron oxide that way is safer, but even the gold is that way safe, but then you have to show that how it is going out of the body. So, that is why excretion of the body is very important to show, and that is why so many systems which are well published and patented and with initial good result, still they are under clinical trial or development, and still they have not come to the market.
But just argue against nanoparticle to begin with are very little in amount, and to make sure they all come out, probably it's a difficult problem to solve, or is it not a difficult problem at all?
Yeah, not like that. Basically, when we talk about these the ADME, as I told you, the distribution, which is we say sequestration into the different organ, because particle cannot freely move into the blood or fluid system. So, obviously, it will be sequestered in some organ, and after sequestrating in that organ, then they have to come out from their organ, and it has to come out of the body. Now, it may not be possible to show in a particular time point that whether all these particles have gone out or from the body or not, but for a degradable system there is no issue. But for all non-degradable system, always we have a query in our mind that whether in the long term they can produce some amount of side effect or toxicity or not. And to complicate the issue more, we know that these particles have a property of translocation. Say for example, initially they are sequestrated in the spleen and liver, reticuloendothelial system, and after some time some of them can translocate in the lung. And even if you have a bad luck, some of them at a particular stage can go into the brain also. So basically these kind of movement issues are also there, which is very difficult to crack. But overall, when we are doing the safety studies in different animal models and other systems, obviously these are the issues has to be properly looked into.
Wow, actually that's very good to know. And so I'm wondering, can nanoparticles be used in thermal therapy for cancer treatment? Is this true?
Yeah, it's a very important area, I feel, because the cancer cell killing, we have got now mainly two direct modalities. One is chemotherapy, where cytotoxic drugs we use to kill cancer cells. Other is radiotherapy. Radiotherapy, basically, to explain in layman's term, it is you are burning these cancer cells. Now, radiotherapy has got a lot of side effect. It also caused a lot of DNA damage. So it has got the radiation associated the cellular injuries in normal cells around these cancer cells also. So and not all cancers also you can use radiotherapy in sensitive areas. So whereas if you can get same effect by hitting these areas of cancer cells, which we called photothermal therapy, then all the side effects of the radiotherapies are gone, and there will be no systemic side effect also. Radiotherapy has got a lot of systemic side effect. So that's why the nanotechnology is really that way very good to really translate into as a photothermal therapy. It's very simple, the scientific principle. There are several metals, particularly gold, which has got the surface plasma resonance, that is SPR. And when you irradiate with a particular laser light, and usually we use the laser of near infrared laser, which doesn't produce much heat otherwise, but when it falls in on these the gold nanoparticle, and particularly the gold nano rod, they have got aspect ratio which produce maximum SPR effect, and thereby they can produce maximum heat. So they can produce the heat in the range of whatever you want depending upon your designing, but usually we want around say 50 to 60 degrees Celsius. So then that area of these the tumor, if you can impregnate them into the tumor, this tumor can be, but the problem is that if you introduce these gold nanorod inside the body to kill say deep seated tumor, even for breast cancer or liver cancer, then you have to show that how they are going to come out. So that's a very difficult thing to do.
Even after they've done the job,
you have to take them out. So that's why I believe that though there is a lot of potential for photothermal therapy, but for deep seated cancer, that's why it is still, its use is yet to be proven safe. But for other kind of use, surface use, it is obviously very perfect situation, one can use it.
So obviously it's a therapy for future, right? Near future.
Yeah, I mean.
Dr. Dinda, your work is magnificent and very applied, and it's within the reach of patients. So we were wondering how long does it take for an idea which is working really well, and you've talked so many about with us today. So how long does it take for an idea to go from lab to the market?
Yeah, to tell you very quickly, I think whenever we first do the proof of the concept, in the proof of the concept study, we do it at the cellular level, at the molecular level, then we do a preclinical study. Preclinical study we do in the animal model, usually the disease animal model. The disease is similar to the human conditions. And with that data, obviously then we have to go for the human clinical trial. There are a little bit of difficulties so far as the nanosystem is concerned. The one is that the moment we say that we want to use it for delivering of certain drugs, then the PK/PD, that is the pharmacokinetics and pharmacodynamics of the distribution of these drug molecule, it gets altered when we load them in the nanoparticle. So basically the PK/PD determination is becoming very difficult in that situation. So it becomes a IND again, the investigational new drug. So that is one of the real block, and there is till date no definite regulatory regulations available for nanoformulations. So that is one the problem we face. Second point is that then when we do the sequestration study in animal, we can show them by in vivo imaging, or you can get samples and take out this drug from the liver, spleen, brain. But in human clinical trial, you can't do that. You will not be able to do that. So basically, indirect projection system we do. And then at the same time, whenever we are using these carrier, so carrier property has to be also, and safety has to be also properly documented. So as a whole, that's why there are several steps before we can bring in the market or for clinical applications. But I strongly feel that if you are using a biodegradable system, you are using the biopolymers which are US FDA approved, and many of them are also in use in different situations. Say for example, polycarpolactone is used for different kind of processes, and so if you are using this kind of material, then if we develop something, it may be easier really to go for clinical trial. But obviously, so there are I think several years is needed before one can idea to product development. Yeah.
In relation to that, are there other examples of successful nanoparticle based drug delivery system available already in the market?
Yeah, in that I think I can mention the two important drugs. One is that Abraxane, which is the paclitaxel the loaded in the albumin nanoparticle, and it is freely available throughout I think the world. In fact, in India also there are a lot of units which are using it. So that's a good example of that. Other is that Doxil, which is the doxorubicin loaded nanoparticle, which is I think coming into the market very shortly. And there are some other countries like Korea, they have got more these systems which are available for the local market. But I feel that a lot of these will come gradually in the market.
So from what you've told us, seems like the nanoparticle based drugs are given either by intramuscularly or intravenously. It might be discouraging for patients because some of us are scared of needles, and in spite of being excited about technology, we might not be excited about the needles. So is there a way you can develop, or there are nanoparticles which can be used needle-free manner?
Yeah, you were, I think, rightly pointed out, a huge scope for application of nanotechnology to convert some of these applications orally. Like to tell you the example is that if you search the literature, there are a lot of anti-cancer drugs which are highly toxic. But now with the help of the nanotechnology, it is possible to give them orally. In fact, they are patented also, though the safety issues and the preclinical trial in animal models are going on in many of the systems, but still they have got a huge potential, so that the chemotherapeutic drugs which are always to be given in IV, they can be transformed into oral formulations. And also many of these vaccinations where we inject them, and these can be have a transdermal needle free injection where this particle can really go and have a much better effect than even the conventional vaccination. So there is a possibility in that.
So technology this good must have used outside laboratory. We have some of us already used nanodots in confocal imaging. Are there examples of nanoparticles in day-to-day life that we are not aware of?
Yeah, there are plenty. In fact, there are fascinating applications of the nanotechnology in our day-to-day. I mean, the whatever you are using. I'll give you some example. You have now the nanoparticle impregnated the textile, and these are the dust cannot accumulate on them, and also the water cannot make them wet. So the ties are freely available, but then if you search the internet, you can get also many other apparels. And then also in cosmetic, particularly the sunscreen, they have got the nanoparticle base itself, and in shampoo they have got anti-infective properties with silver nanoparticle they are using. And in fact, apart from that, there are certain glasses also where it has got a sensor of the light according to the glass changes. So there also the nanoparticle based system. The carbon nanoparticle and nanosystem is used for reinforcing steel. Carbon nanorods are also used for different kind of very light composite material. So there is a huge application of the nanotechnology in the different kind of industry.
So technology is exciting. One would wonder how old is this technology?
Yeah, that way the very interestingly, this technology really came into the mind very accidentally. The Feynman, Professor Feynman, who was a Nobel laureate in physics, he was invited to give a talk in Caltech, in California Institute of Technology. The title of the talk was "There is plenty of room at the bottom." And the talk he gave 1959, and this was published in his excerpt of the talk is published in 1960. So there he hypothesized or postulated that is it possible for us, say for example, write the whole Bible on the top of a needle, tip of a needle. So and then he went on saying that we can miniaturize our instrument, our motors at the nano level. And so these all molecular interaction and all the functions can be altered, and so that was the thing. And he also told that it can be used in different systems, including the biomedical system, in engineering system, and everywhere. Yeah, after that, I think after 1960, till 1999, almost there was hardly any activity. Apart from there are some research by IBM and some other group. Maybe MIT also worked on some, but then after that in 1999 the Freitas published a book on nanomedicine, but many of his were more of a postulation with little bit of data, and there he told that these really nanotechnology, if you want to apply in medicine, then it can do wonder. After that, it's I think 2002 is the landmark year for particularly application of nanotechnology in medicine, when NIH has developed a 5 years project on application of nanotechnology in medicine, and they form a huge collaborative group across the different institute in USA, and they really form a collaborative group among engineers, doctors, basic scientists, chemists, physicists, and computational biologists, computer person, mathematics, and so on, and including the statisticians, statistical bioinformatics. And then these actually led to real the thrust in the area of nanomedicine research. And after that, if you can see, there is huge number of articles, the journals. I think as a single area, the nanotechnology, including nanomedicine, is the area where maximum number of journals and publications are available. If you see as a particular topic of science. So it's that way the journey. And then now, if you see the volume of the money invested and research which is coming up in nanotechnology, it's really progressing and increasing.
Dr. Dinda, you have achieved success in demonstrating potential of nanoparticle-based drug delivery system. But what's the overall standing of Indian science in this field?
No, the nano research in India is also huge, because if you see there are almost 19 or more the advanced facility or center of nano research funded by DST. There are several other institutes like Indian Institute of Nanotechnology is being developed. So if you see the volume of publications and research data, is really great. But one thing I think I can mention, that so much of publications and ideas are coming up from the different institute, but actually its translational level is, I think, not satisfactory, because the number of product which is coming out of Indian nanotechnology research, I don't think yet we can name much around that. So there is a gap which is there in this area. Otherwise, I feel the milieu of nano research, nanotechnology, nanobiotechnology research in India is really quite vast, and they are doing pretty well. Very specific application in medicines, I think we are lagging a little bit, and I feel with time it can improve, and it will improve.
And just additional question to that, what does it take to survive and thrive in Indian science? If you can give some pointers to young scientists, students who are probably very keen to know the mantra of success.
I think the mantra of success is your real tenacity and perseverance, because here you have to always think of that failure is always your way. And because of there are several issues, like you can't imagine to get a lab, say for example of Harvard or MIT or UCLA in even many of these institutions, and there are few really that kind of hi-fi institute where everything is available at your hand. You have to see our financial economic situations, and we are still a developing country. Yeah, even if I have mentioned that there are quite a bit of infrastructural problem in India like many other developing countries, but that actually help you to think more, because I have worked in US system, in Japan, in Australia, obviously different countries. I realized that if we have got always a paucity of our resources and also facilities, so we think much more. We try to do few experiments which we feel that they will be much more successful. So that way our thinking is more, idea mapping is more. So it is that's why giving us not very bad result. We are doing pretty well if you compare, and in that way our research is more competitive financially or economically. So I think this strength we should utilize, and so that way if you do that, if you try to do shortcut, then there are side effect of shortcut also. So I also know that, because internationally we cannot do much shortcut in different areas. But so far as in Indian science and the younger scientists is concerned, that's why I feel that they should be always keep a goal that even if the failure comes, but I will really pursue and try to work out more and more. And then I think definitely there is a great hope of our very young and talented brains.
That's actually a very valuable suggestion you've given us. So I'll wrap up with asking a question, more sort of a personal question. So you're obviously very busy with your research, mentoring students, and also probably involved in administrative duties at AIIMS. So we are wondering, does all this leave any free time at all, and how do you balance work and life, or there is no balance for a busy scientist like you?
I think it's a very important question. So, to tell you frankly, like I always try to make a balance, because I realize that whatever you do in your research domain, in your the medical practice domain, diagnostic domain, and at your home, see everywhere there are certain things which are essential. You cannot avoid that. So I personally feel that yeah, sometimes my research pressure is too much, but then I have made a habit of thinking things, even the research ideas, while I am doing something, and even watching movie or listening to music. And but then cutting time from the family is sometimes very difficult. So always I try to allocate some of the time to my family. So basically sometimes I compromise a little bit of cutting my time from science and then spending with my family, and sometimes reverse. So in always you have to have problem, but more and more I am getting involved with a lot of policies and this and that. I agree that it is becoming pretty difficult, really challenging to maintain. But I think I enjoy every moment. That way, whatever I do, I try to enjoy. So that gives me a pleasure to enjoy my work, enjoy my family, and enjoy whatever I am doing.
Dr. Dinda, thank you so much for sharing your experience of living science. It is very exciting to know that nanoparticles can be used in treating infectious diseases, cancer, and also has a great potential in be a main component of single shot vaccine. Dr. Dinda, thank you so much for being with us. We really appreciate you giving us time, and again, thank you so much for being with us.